So this is the last video on our discussion of enzyme activity, and we're going to start taking a look at the things that can affect enzyme activity that I mentioned earlier. Enzymes are active only over a limited pH range, and most have a specific pH at which their catalytic activity is optimal. And this graph or figure shows us 4 different enzymes, and we'll look at that in a moment with respect to their activity. But you have to keep in mind that enzymes are proteins. They have active sites, and there may be ionizable side chains in those active sites that are involved in interacting or catalyzing that reaction. Also, substrates may have ionizable side chains in their amino acids that might be critical for interacting with the enzyme. So pH is critical, depending and covers a small pH range for most enzymes. However, as we can see from this graph, not all enzymes function optimally at the same pH. If we look at pepsin, which is a gastric protease, its optimal activity, as you can see here, is between 0 and 2. And that's because the pH in our stomach is around 2. So pepsin functions at low pH. Trypsin, we looked at trypsin, or used trypsin, I should say, when we were analyzing the sequence of proteins. Remember, it is a protease. It cleaves proteins at specific sites. It has an optimal pH of around 7.4. That is just slightly basic. Here are 2 others, papain and cholinesterase. Cholinesterase actually is pH sensitive below 7, but not between 7 and 10. So that's this curve here. And this one is relatively functional between 4 and a little less than 9. Trypsin, I didn't mention this, it's an intestinal protease, so that's why the pH is slightly basic here. It's in an alkaline environment. So pH then is going to affect the activity of enzymes, and it also may affect then the Km, Vmax, or both. Enzymes are also temperature sensitive. Rates of enzyme catalyzed reactions generally increase when increasing temperature. However, above 50 to 60 degrees, enzyme activity declines. So we have function at lower temperatures. However, there is a maximum temperature at which may and can denature that enzyme and make it non-functional, period. It is common to store enzymes in a freezer, and when they get warmed up, they go right back to work. However, if it's become denatured, you're out of luck, basically. So enzymes may be inhibited reversibly or irreversibly. Well, and we'll take a look at that, that phenomenon. Reversible inhibitors may bind at the active site or at some other site. Enzymes may also be inhibited in an irreversible manner. We'll look at an example of that. And actually, penicillin is an irreversible suicide inhibitor, and we'll look at that too. So covalent modifications I mentioned as one way that enzyme activity can be affected. Although some covalent modifications are permanent and inhibit the enzyme irreversibly, other covalent modifications are reversible, and that's what we're looking at right here. And we will see this in one of our pathways. So this is an interconvertible enzyme. So we have enzyme with a hydroxyl group, and this is its active form. It can be phosphorylated. So here we need a molecule of ATP, and we need an enzyme called a protein kinase. This protein catalyzes the transfer of a phosphate group to another molecule. In this case, it's the enzyme. And then ADP is released. This enzyme, when phosphorylated in a covalent modification, is inactive. Phosphorylation can activate. It depends upon the enzyme. And we'll see examples of that too as we look at some of our metabolic pathways. Again, this is reversible, excuse me. To reverse this, we need a protein phosphatase. And this enzyme is responsible for hydrolyzing that phosphate group and making that enzyme active again. I mentioned also zymogens. These are inactive precursors, or another word for them is proenzyme. And they require a proteolytic cleavage to produce that active enzyme. So what we're looking at here for an example is proinsulin. It's an 86 residue precursor to insulin. There's a proteolytic cleavage that occurs between, or at, I should say, residue 31 here, or in between 30 and 31, and another at 65 here. And what that does is leave, so this blue segment is released, and we're left with this A chain, which is residue 1 through 30, and I'm sorry, this is the B chain. B chain is 1 through 30, and the A chain is 66 through 87, I believe it would be. These are linked by what? Disulfide bridges. Okay, we've seen those before too. So part of their tertiary structure is due to those disulfide bridges. I'm tongue-tied today for some reason. Here's another example. Another example of a proteolytic activation. We're not looking at. We are looking at an inactive zymogen. So chymotrypsinogen is the precursor to chymotrypsin. We've also used that in digestions when we analyzed protein sequences. So here's our inactive zymogen. It's 245 residues. It's cross-linked. These yellow lines are representing disulfide bonds within that zymogen. The protease trypsin cleaves this zymogen at arginine 15. Okay, so we see that here. So there's a cleavage. We're still being held together by disulfide bonds. Now, once that cleavage occurs at Arg 15, we have an active enzyme, and it's called alpha chymotrypsin. This enzyme acts on other alpha chymotrypsin enzymes. So, in a sense, it's a self-digestion, and we have a cleavage at Lu 13 and tyrosine 146 and asparagine 148. So then we have the release of a CR-ARG segment, dipeptide, and a threonine asparagine dipeptide as well. So what we're left with is we've moved a sequence here and here, and we're left with the mature protease alpha chymotrypsin. So it's composed of 3 peptide chains, A, B, and C, that remain together because they're linked by those disulfide bonds. Another example of a cascade, or this involves a cascade leading to blood clotting. There are 2 pathways by which this cascade can be activated, and typically both are activated by tissue damage. So the intrinsic pathway occurs within the damaged tissue surface is detected, which sets off this cascade, and we go down from 12 down to 10. Again, this activates this activates this one is an activator of this, so this is the cascade. Also the extrinsic pathway, we've got trauma on the exterior, we activate 7, becomes active 7A, which is tissue factor, and it also activates X, or factor X, to its active form, and then they both, from this point on, they converge. Activation of prothrombin to thrombin, fibrinogen to fibrin, and then we result in the clot formation. So formation of blood clots then is the result of a series of zymogen activations. Okay, so now we're going to take a look at inhibition. So we're going to have an inhibitor that's going to affect our reaction. So here's our Michaelis-Menten equation, enzyme plus substrate in equilibrium with our transition state, and our K2 going to product plus the enzyme. If we have an inhibitor present, we are going to also have that EI equilibrium plus substrate, but no reaction. So the reaction is inhibited. And we're going to look at 3 types of inhibition. So the first is competitive inhibition. So the substrate and the inhibitor compete for the same site on the enzyme. So here we're looking at the Lineweaver-Burke double reciprocal plot. We have the uninhibited reaction, or enzyme activity. This is plus inhibitor, and 2 times the concentration of that inhibitor. So what you should notice about this is 1/V max does not change. That's unaffected by the inhibitor's activity, or presence, I should say. However, minus 1 over the Km does. So the Km apparent is increasing, because as you increase inhibitor, that value decreases. So that reflects an increase in the apparent Km. This can be overcome, and we can do that by increasing the substrate concentration. Pure, non-competitive inhibition. So this is a case where the inhibitor binds to a separate site on the enzyme. Order does not matter here. So what I mean by that is we have enzyme, and it can be bound by the inhibitor, or we can have the ES complex. It can still be bound by the inhibitor. Same outcome. So here is minus inhibitor, or no inhibitor, plus inhibitor. Minus 1 over the Km remains the same, or the intercept with the 1 over S axis does not change. However, there is a change in the intercept on the y-axis. So this tells me that Vmax is decreasing. So I is decreasing the amount of the active enzymes, or enzyme present. So Km is unaffected because the uninhibited free enzyme is still capable of normal catalysis, but the Vmax is decreased because the inhibitor lowers the active enzyme concentration. And finally, uncompetitive. So you need to be able to recognize and discuss the differences between these types of inhibition. I will give you an exercise in which you can practice that, hopefully without referring to your notes. So get a good handle on these types of inhibition. Now, looking at this Lineweaver-Burke double reciprocal plot, we see parallel lines. Okay, so I call this pun competitive to remind myself that these are parallel lines. So what does this graph tell us? Well, obviously the slope is constant. Both these lines have the same slope. Here's minus inhibitor, here's plus inhibitor. But Km and Vmax both decrease. Okay. So Km, Vmax are decreased. So I mentioned penicillin when we started this section on the things that affect enzyme activity. Penicillin is an irreversible inhibitor of the enzyme glycoprotein peptidase. This enzyme catalyzes an essential step in bacterial cell wall synthesis. So this is why penicillin works on bacterial infections, unless that particular bug has become resistant. So suicide substrates are basically inhibitory substrate analogs. So they look like the normal substrate. However, the reactive groups in the analog form covalent bonds with the functional group in the active site of the enzyme and render it non-functional or inactive. Sometimes these suicide inhibitors are called Trojan horses as well. So we talked or mentioned that pharmaceutical industries manipulate and try to create analogs to treat and hopefully better some of the conditions some of us may face. And an example of a drug that they designed, which most people are familiar with, Viagra, ended up having an unexpected outcome in the program. They originally thought that or tried to produce a phosphodiesterase inhibitor to treat angina and hypertension. So how would this work? Cyclic GMP, which is shown here on the left, is associated, and I'm not going to go through all the mechanism, but the presence or the concentration of cyclic GMP is associated with relaxation of smooth muscle, which, if this happened in the heart, would lower blood pressure, heart rate, relax that heart, so you could help with that angina. Viagra is basically a phosphodiesterase. The phosphodiesterase cleaves the cyclic GMP to form GMP. So it's no longer cyclic or 5' GMP. When that happens, that initiates muscle contraction again. So the thought was if we can inhibit that phosphodiesterase, we're going to keep the concentrations of cyclic GMP high, and then vascular muscle relaxation, so forth. It worked, but not in heart tissue. Some of the men involved in the clinical trials reported differences in their function as far as penile erectility, and thus it is now being used for that purpose. I asked a question, are all enzymes proteins? And no, they are not. We're only going to look at this just briefly, and in fact it's just this slide. Ribozymes, segments of RNA, that display enzyme activity. We will look at some ribozymes in I think actually the last unit that we look at the end of this semester. Abzymes, antibodies that are raised to bind transition state of a reaction of interest. And if you want to take a look at or read up on that, this was a very good review. I know it's a little old, but it is a very interesting review of abzymes. So is it possible to design an enzyme to catalyze any desired reaction? So you can engineer enzymes by mutagenesis in a test tube and replace active site residues with new ones that might catalyze a desired reaction. We aren't going to get into much, really any discussion of how that might occur as far as the technique goes. But other approaches design totally new proteins with a desired structure and activity. Protein folding and stability issues make this approach more difficult, but there has been some good work done on this. And you do begin by using computer modeling. And some of those websites that I listed when we were discussing proteins would be helpful in this case. Cellular environment may provide complications not apparent in computer modeling. So there's a lot more at stake and a lot more work involved with totally designing a new protein versus some kind of mutagenesis. And that concludes our discussion of enzymes. Up next will be carbohydrates.